presented by Caltech.
Good evening, everyone. Welcome to this lecture in the William and Myrtle Harris Distinguished Lecture Series in Science and Civilization. This lecture series began in 1996 and has been supported through the generosity of William and Myrtle Harris. These lectures are run through our program in History and Philosophy of Science and designed to bring people to campus from a variety of fields, historians, philosophers, scientists, researchers in other fields, authors. And the theme of these lectures is the broader place of science in society, exploring the historical, the social, cultural, and ethical dimensions of science. William Harris received his Bachelor's of Science degree and his Master of Science degree in Mechanical Engineering at Caltech. Bill and Myrtle were supporters of Caltech in many ways, longtime members of the Associates, members of Caltech's President's Circle. They have contributed to the Summer Undergraduate Research Fellowship Program and to the undergraduate scholarship programs. So this is really a wonderful series. We're very grateful for their support for this series. Tonight we're very pleased to have as our lecturer Dr. John Ioannidis. Dr. Ioannidis was born in the US but raised in Greece and got his bachelor's degree at Athens College and his Doctor of Medicine at the University of Athens and did his residency at Harvard and then went back to the University of Athens to get a proper doctorate. We get to say who is and isn't a real doctor. He's been affiliated with many wonderful institutions, Harvard University, Tufts University, Johns Hopkins, the National Institute of Health. And then for a little over a decade, he was a professor in the Department of Hygiene and Epidemiology at the University of Ioannina in Ioannina, Greece. He moved to Stanford in the School of Medicine in 2010, where he is now. He is the C.F. Renberg Chair in Disease Prevention and holds a list of titles that are so long that if I read them, he wouldn't have time for his talk, but he lists a few of them here. He's a member of many scientific academies, including the Association of American Physicians, the European Academy of Sciences and Arts, and the European Academy of Cancer Studies. He is one of the most prolific, influential, and widely cited researchers in any field of science. He has authored and published over 800 scientific research papers, and he's worked on a variety of topics, but the research he's going to talk about tonight and sort of what put him on our radar for this lecture series is his research on research, so his research on the nature of scientific research and his research into how, when, and why science works and doesn't work, ask questions such as how often are the findings in scientific research papers successfully replicated, how often do replication attempts fail, what are some of the factors that lead to findings being reported and published that turn out later to be incorrect, and perhaps most importantly, what can be done to make scientific research more reliable and more efficient. Perhaps his best known paper has the provocative title, Why Most Published Research Findings Are False. This was published in the online Public Library of Science in 2005 and is the most downloaded paper in the history of that publication venue. It's been accessed 1.5 million times. You can't do better than that without a picture of a Kardashian. He's going to speak to us today about empirical research on research and the reproducibility crisis. Please join me in welcoming Dr. Ioannidis.
Thank you for the very kind invitation. It's a great pleasure to be at Caltech, and thank you all for coming in what is a late evening lecture. I will try to share some thoughts and hopefully leave some time also at the end for questions and discussion. So it's a very exciting time for doing research on research, and the main reason for that is that we have lots of data. Science has been very successful. Just looking in Scopus over 15 years, there's 15 million scientists publishing in the world of science. And Google Scholar probably catalogs something like 160 million papers. Each of these papers may have actually many analyses. Some papers include a background of millions of analyses that have been done. Sometimes all of them are visible or referred in supplements. Other times some of them are visible, and it's hard to say how many others have been done. Clearly, this is a success story, but at the same time, it means that we can start asking questions about how much of that volume is true, significant, important, makes sense, takes us to the next level of translating that knowledge. Now, even though this is a huge galaxy, this is the galaxy of science, this is some work that my colleague Kevin Boyek has done for the map of science. It's 20 million papers out of the 160. It's fragmented. There's lots of fields and subfields and disciplines and areas that are created, grow, expand, merge, split, sometimes dissolve. So in these 20 million papers and 2 million patents, there's about 200,000 clusters. Each one of them lasts anywhere from 2 to 16 years. 16 is the largest follow-up for these type of analysis here. Obviously, some can go even longer. Each of these disciplines may develop its own rules on how it uses the scientific method. We all use the scientific method, but how exactly we are using it and what we prioritize in using different aspects of the scientific method can vary quite a lot. We are all seeking significance, but the problem is that almost all scientific papers that you will see, almost in any field that you might be working on, are statistically significant. Recently, we published a paper in JAMA where we text mined the entire PubMed biomedical literature of millions of papers over the last 25 years and close to 1 million full text articles. 96% of these papers, whenever they had a p-value, it was a statistically significant p-value. Almost everything is significant. Every time I try to convey a message, I'm trying to say that I'm doing something significant. You know, invited me to give you some significant insights, not something that you would waste your time. All of us are trying to do this, but how much of that is really significant is a very different story. This is the data from the PubMed analysis, looking at PubMed and PubMed Central. It has a full text. The rate of significance actually is going down a little bit from 98% to something like 95% over these years. But still, it's very high. And what this means is that yet another significant discovery is boring. You know, you just see of those every day. I want to see more negative results. You know, I want to see perhaps both significant and non-significant results, but what I want to see is trustworthy results rather than just nominally significant ones. Why do we get this epidemic of statistical significance? Much of that may have to do with how we perform research. What is the typical recipe for performing research in most disciplines? Most disciplines have promoted the notion that you need to be a principal investigator. You need to bring funding and resources to your team. You need to be competitive. You should be very careful with how many other people you collaborate. If you give away your data, if you give away your ideas, then maybe you will not be able to get funded next time. This means that most principal investigators are working with relatively small sample sizes. They're working largely in silo in a very competitive environment. They need to get significant results so that they write up their next grants, that we found something and we want to pursue that further. There's lots of disincentives towards sharing. There's disincentives towards pre-registering ideas. There's incentives for allowing ourselves to do more post hoc exploration and come up with significant results all the time. Also, there's no incentive for replication because you do a lot of work and you come up with something that looks interesting. Why would you replicate if no one is asking you to do that? If you replicate, there's a very high chance that this will not pan out. And all that work then will have a lot of difficulty to be published in many of the prestigious journals, or all of the prestigious journals that probably would want to see some significant results. So recently there has been a lot of attention given to reproducibility because we increasingly recognize that much of that cloud of significant results is actually not true. They're false positive or spurious findings that cannot be replicated. They cannot be reproduced. So in the last several years, there's an exponential number of papers across all scientific fields, some fields probably more than others, but practically across all fields that focus on reproducibility of results. And this is a search that I have used along with Dan Fanelli and Steve Goodman, different terms like research reproducibility, reproducibility of research, reproducibility of results, reproducibility of study, and so forth. There's many more that actually pertain to these same notions that don't use that exact terminology. But it's showing the very rapid increase of that literature. Now, what is reproducibility to start with? There are different types of reproducibility. There's reproducibility of methods, there's reproducibility of results, and there's reproducibility of inferences. So reproducibility of methods is something that is very familiar probably to people in the computational sciences, like computer science, people who work with software, with code. If you write some code and you run it through some software, you want to be able to get the same result. Now, or almost the same result. It could be a simulation that has, let's say, Monte Carlo iterations, and maybe you get slightly different result if you run 1,000 Monte Carlo simulations a chain, another one downstream. But it should pretty much give you a very consistent conclusion. Reproducibility of results means the ability to produce corroborating results if you do yet another study. Not the same sample, not the same simulations. You go to other participants, to other observations, and you want to make sure that you get the same conclusions, the same inferences at the end of the day. And reproducibility of inferences, it means that you get some results from 1 or 2 or 10 studies, and you have 2 or 3 or 10 experts, and you ask them, what do you make out of that? Maybe exactly the same results, but they may interpret it in very different ways, depending on what they believe, what their background is, what their perceptions, their priors, their biases might be. So we need to differentiate between these 3 types of reproducibility. We also need to take into account the fact that different fields have different parameters that make sense to them when they're talking about reproducibility. So here's a list of these differences that could affect what people mean by reproducible research. The degree of determinism. Physical science is probably, even though one might say that there's also lack of determinism even in physical sciences, usually there's more determinism sought in the results compared to what you would expect in social sciences or biological sciences. The signal to measurement error ratio could vary a lot. The complexity of designs and the measurement tools. The closeness of fit between the hypothesis and the experimental design data that we say that's good enough. We don't worry about rejecting the speed of light. I mean, if you run some statistical tests on measurements of the speed of light, maybe you may reject the null hypothesis that there is no difference. But then you say we don't care about that because this is within the noise that we are willing to accept in this type of measurements. The statistical and analytical methods to test hypotheses. The heterogeneity of experimental results that is considered to be typical for the field. Some fields do not want to have any heterogeneity. Other fields say, oh, that's okay. I expect to see diversity. These are different people, different populations, different settings, and so forth. Whether there is a culture of replication, transparency, and accumulating knowledge in the field or not. Some fields perform replication studies, others don't want to hear about it. They say that this is Me Too research, that's a waste of effort, I want to move forward to the next experiment that it will be completely different. What are the statistical criteria for truth claims? Many fields are happy to get a p-value of less than 0.05, which usually, if you interpret that with some Bayesian reformatting, it means that they're happy to get 1% chance of being correct. But you know, and they either know that or they don't know that. Other fields are asking for a p-value of 10 to the minus 8. Like genomics, standard, you need to go to a genome-wide significance of 10 to the minus 8. Many databases that I use almost on a daily basis that use population-level data from entire countries, I get p-values of 10 to the minus 100. I believe none of them, and none of them is probably reflecting a true signal with very, very few exceptions. So what are the thresholds that are considered to be good enough? Can vary a lot. And the purposes to which findings will be put and the consequences of false conclusions. So is that just a curiosity that it's me and another 3 people working in that field? We may be misled, but we'll find out down the road and we'll correct our error. Or is it something that is a new drug that will be used by millions of people, and if it doesn't work, if it has severe adverse events, people will die? There's lots of grading in the consequences of irreproducibility. Allowing for that diversity of reproducibility, we have evidence increasingly, empirically, from a very large number of fields that there is some reproducibility problem, or at least we have plenty of room to improve on our reproducibility performance across very different fields. This is an empirical assessment looking at fMRI data for association between specific foci in the brain with different tasks or phenotypes of interest. And it's about 1,800 data sets. You see that the only way to discover a very large number of foci in this field is to perform very small studies. If you start performing larger studies, you never get a very large number of foci. And we see that also in meta-analysis. If you perform meta-analysis, that you join multiple data sets and you standardize the analytical approach and you make everything similar in the way that these data sets are analyzed, the number of foci tends to go down with larger and more robustly controlled and analyzed data sets. Not just a problem in fMRI studies. A couple of years ago, along with a number of colleagues, we looked across a wide range of fields in neuroscience, which is a very vibrant and active field, and lots of brilliant people are working in neuroscience. And we found that there's some common denominator, regardless of whether we're talking about fMRI or voxel morphology or other neuroimaging studies or animal studies, experimental studies, cell culture studies, signaling studies, cell biology studies, preclinical experiments, clinical trials. The common denominator was that most of that research was underpowered, had very small sample sizes, was underpowered to detect what would have been modest and meaningful effects that one would like to pursue further. When a field is operating in that environment, which is probably the consequence of the PI prototype that has a limited budget and can only come up with limited sample size studies, in that environment you expect to get tons of false positives and tons of false negatives. And you don't really know which one is the most important problem. Sometimes missing true effects is a major deal. Sometimes coming up with spurious false positives is also a major problem. Here's a completely different field. This is economics. This is observational economics. And even though it's so far apart in that galaxy of science that I showed you up front, if you plot economics versus neuroscience, they would be many light years apart. They share the very same fundamental problem. They're both underpowered fields, meaning that most of the studies that they do are underpowered to detect modest effects that might be of interest. This is an analysis that I have done with Tom Stanley and Christo Kouliagos, who are both economists, and looking at every topic that we could identify in economics. We came up with 130 topics that had a meta-analysis, and that includes more than 10,000 studies with more than 70,000 estimates of effects. You see that the vast majority of these studies are underpowered to detect what would have been modest effects. There are a few fields where we have the benefit of replication being very easy to do. So as I said, many fields don't do replications because why waste our resources? You know, I'm a poor PI, I'm just trying to run the best possible studies and move to the next target, and even thus, I can only run small studies. But there are a few fields where replication has been very inexpensive or has become inexpensive. And also there are some fields that realized early on that replication was important because whenever someone was trying to replicate something, nothing seemed to pan out. So people started saying, well, we'll better be careful on what we publish. One such field is genetics. Replication became a sine qua non in the last 10 years because we had multiple advances. We had platforms that could capture the genome in much better coverage. The price of measurement went down about 100 million-fold over 10 years. So for the same price that one could genotype a couple of single nucleotide polymorphisms in an individual or two, now you could just do the whole genome in several thousands of people. And then people realized that this is an important thing. And once replication became a sine qua non, we could go back and reevaluate our previous literature of tens of thousands of patients of genetic associations of disease that we had found in the previous era when people were targeting one gene, one or a few variants, and one phenotype, coming up with a nominally significant result and publishing that as here's an association of a disease with the genetic. When that was done in large scale, only 1.2% of that literature of targeted hypotheses, candidate hypotheses of the past could be replicated with a new wave of massive replication with very large sample sizes and with very standardized methods in large consortia of investigators combining data across multiple teams. So, for example, in 2006, the best review in the area of smoking had concluded that we know 359 genes that are related to smoking behavior. Then when we start performing consortia with hundreds of thousands of individuals at some point, only one of these 359 gene variants could be replicated. Now we have about a dozen variants that we know that they are associated with smoking behavior. We have less knowledge, or quote unquote, compared to what we had in the past, but now it's far more rigorous and we can replicate it repeatedly across multiple teams. Another field where replication became very popular in the last 5 years is preclinical research. And here the champion has been the industry. This sounds a little bit weird because usually we hear about the industry trying to hide things under the rug and not willing to do lots of testing that would rock the boat. But in that case, the industry was very interested to find out whether drug targets that were being proposed by academic investigators in the best academic centers actually did work or not, because otherwise they would be wasting hundreds of millions of dollars trying to develop these drug targets into drugs to be licensed. So a number of series of investigations tried to repeat experiments that had been published in key journals with high impact factor by the best academic teams in collaboration and in communication with the original authors. And the replication rates in these efforts of reproducibility range from 11% to 25%. So pretty disappointing, but at the same time an eye-opener about how much trust we can place in this research and how can we try to work towards improving that rate of reproducibility. There's many other fields that have signals of reproducibility crises or problems. They range from neuroscience, which I mentioned, pharmacology, genomics, bioinformatics, stem cell biology, in vitro testing and oncology, chemistry-led discovery, computational biology, pathology and biomarkers, organizational psychology, observational research in general. I'm just going to show you one more example from psychological science. About 4 years ago, I published that paper trying to summarize a number of papers in a whole issue of Perspectives in Psychological Science that combined empirical studies on research in psychology. So the field had started questioning itself and had started going some soul searching about, do we need more replication to be done and what are we doing? Are we doing things right? So my estimates, based on preliminary observations, were that there are 6 patterns that you can have based on what the discovery and the replication results show. And the frequencies were as follows. Correct, meaning that optimal, meaning that you get a correct discovery and then you replicate it and you get it again. That's less than 1%. Self-correcting, I got it wrong, but then I did the replication and I corrected the error, less than 1%. False non-replication, I found something important, but the replication didn't work for whatever reason, less than 1%. I got it wrong, and I tried to replicate, and I had the same errors and the same biases again, so again, I got it wrong. Second time, that's perpetuated fallacy, that's 2%. 43% unconfirmed genuine discovery, which means that I found something, but then nobody replicated, didn't even try. And 53% unchallenged fallacy, which means I got it wrong, and nobody dared to try to see if it is correct or wrong. In last fall, there was that paper published in Science with the results of the open science collaboration. 273 psychologists and their teams tried to replicate 100 high-profile papers published in high-impact journals in the field, and they could reproduce about 1/3 of them. With different definitions of what is reproducibility, that exact proportion may vary, but roughly 2 out of 3 could not be reproduced. And also the effect sizes were far, far smaller. So these are the effect sizes in the original studies, and these are the effect sizes in the replications. The replications having about 1/3 of the effect size compared to the original. This is a very nice study in economics. The lead author actually is from Caltech, and doing the same thing with 18 studies in experimental economics. And 2 out of 3 in that case could be reproduced. The sample is pretty small, so one might say that economics is doing a little better compared to psychology in that regard, but obviously one would like to see more data. Is there something different about experimental economics compared to observational economics that I showed you earlier? Well, there's many things. I mean, first of all, it's experimental. Based on theory, what I had suggested in my PLOS Medicine paper in 2005 is that randomization experimental design should be a key factor for boosting the reproducibility and the credibility of some research finding. Second, this is a field that has a strong tradition of sharing information, of making things shared among investigators, both in terms of the data and the protocols and how exactly the experiments were done. So this is good news. It means that some fields can achieve substantially better, hopefully, reproducibility rates. Another phenomenon is when you try to reproduce something and you still get some signal, but the signal is much smaller. It's not that it's completely null, but the signal is not really big enough to really be meaningful to take to the next step, especially if you want to apply that information. This is something that we see in many biomedical fields, and this is an empirical example from biomarkers. There's a huge literature on biomarkers, and in this study we focused on the most highly cited papers on biomarker research. We compared the effect size in the highly cited study on the horizontal axis and the effect size in the same scale, relative risk, in the largest study that had been done to date. And you see that almost all of the action is below the diagonal. It's not exactly 1, which is the complete null effect. It's a little bit more than 1, even in the largest replication study, but usually the effect is not big enough that I would use that biomarker for some clinical application. It's the same thing when you go into multivariable prediction models. These are models that may include multiple factors, including multiple biomarkers and demographics and other clinical risk factors. Again, the derivation study tends to get more favorable results compared to the validation study. Most of the action, again, in terms of the area under the curve, which is the discriminating ability of the model, tends to be below the diagonal. Some countries may do worse. So this is some work that I did with Dan Fanelli, where we looked at fields that differed to the extent that their measurements were concrete and objective. So we looked at behavioral and non-behavioral. Non-behavioral was represented by genetics, and behavioral from psychology, psychiatry. And we found that studies from the US compared to studies from other countries tended to have more deviation of their effect estimates. So it could be in either direction, but somehow larger effect sizes were seen when you had behavioral measurements and or behavioral outcomes in studies coming from the US. The explanation that we gave for that was that when you have behavioral measurements, you have more noise in the measurement. You have more noise also in the analytical process. So if you have more pressure to generate an extreme result, maybe you will do that more easily as compared to a genotype that you cannot really modulate. The genotype has an error now of 0.01%. So you cannot really modulate that result as much as how do you feel today? Are you happy? Some psychological aspect of well-being. And maybe investigators in the US have been under more pressure to generate more extreme results because funding is more tightly connected with generating not only significant but also big extreme results. Would that study be reproducible if we were to do it in the near future? I'm not sure, because now there's pressure to reproduce, to produce extreme results probably also in many, many other countries. So I think that the pressure to generate extreme effects may be more generalizable. The advent of big data is adding another dimension of complexity, and this is something that we see very frequently nowadays, claims about making more discoveries. I do believe in big data. I do work with lots of big data sets, but as I said, I come across lots of irreproducible findings. And there's a high risk because we're working with very low priors and in a very agnostic mode and with data of very variable quality that actually the problem of irreproducibility may get magnified in the big data era. Finally, the last confounder that I want to mention before moving into some potential solutions is what I call stealth research. There's a lot of interest in reproducibility and making research more transparent and trying to make sure that we can get the same result if we repeat it or test it again or check it or validate it. But there's also a very strong wave towards making research less transparent. And this is one example. About a year ago, I wrote a paper in JAMA where I mentioned that I read all these wonderful stories in Forbes, Business Insider, Fortune magazine, lots of business-oriented magazines about a company that claimed that they would revolutionize everything in medical testing. They could do that for 100th the price very quickly with high accuracy. They would just take over and disrupt the entire medical establishment. I thought that that was fantastic. So I said, I want to work with these people. They're actually next door to my office almost. And then I searched the literature, and there was nothing published about that. So I wrote that paper about the need to move that stealth research to more visibility. And then Wall Street Journal followed several months later, and the Department of Health followed after that with claims that nothing was reproducible, and there were huge problems and error rates, and people could be killed actually by using that technology. And we're still waiting for some good data to emerge. So some solutions, and I would like to spend more time with you discussing some of these solutions. There are many solutions that are being proposed to these problems, and I have to admit that many of those do not have strong empirical evidence. Some of them do and have worked, and maybe we can see whether we can transplant them to other fields. But many of them have been proposed and never really been very rigorously tested. I tried to summarize those in a paper in PLOS Medicine about a year and a half ago, and I will just highlight some of the key ones. First of all, some fields have recognized that replication is a great thing. I mentioned genomics. Nothing can be published in a major journal if you want to publish on genomic associations of diseases and phenotypes. You need to have replication, you need to have collaboration, you need to have large consortia with practically all the teams or multiple teams working in the same field join forces and publish a single analysis combining all the results and sharing all the data among themselves with a common analysis plan. Pre-registration is another highly debated option. It has worked in some fields like clinical trials, but how can you pre-register things that you don't even know? If you're doing highly exploratory research, what exactly are you going to pre-register? So the solution to that might be that we want to be transparent about what level of pre-registration we're talking about in that research project. Is it level 0, which means that's completely blue sky science. I have no clue what I'm searching for. I'm just trying to collect observations and looking in the dark. If I find something, I let you know. And I will tell you, I had no clue where I was going. Now I got this wonderful result. Please try to replicate it. Level 1 is registration of a data set. So I register that I have that data set with 1,000 variables that I can throw to you at any time with lots of chi-squares or Fisher exact tests, and who knows what we'll get out of that. Level 2 is registration of a protocol. I have a very specific protocol in mind, and this is how I'm going to run it. Level 3 is registration of the analysis plan. I don't have just a protocol, but I know exactly how the analysis will be run. And the same hypothesis, the same protocol can be run in very, very different ways. Level 4, I also give you the raw data. Level 5, it's all in open view as I am doing it. I'm loading my protocols and my plans for today. You can see what I'm planning to do in the afternoon. You can give me feedback. Don't do that. Don't waste your time. I've done that. It doesn't work. Or do it in a better way. How much of that is happening? It's not science fiction. It is happening. We do have lots of interest about sharing and openness. This is a paper that we published 5 years ago now, and we have repeated the exercise recently, looking at the 50 highest impact factor journals across science and whether they have policies in place for sharing information, for sharing data sets, raw data, protocols, materials, and even making that a prerequisite to publication. We saw lots of green color. That's good news. It means that these journals have policies in place to facilitate that and to ask for that. However, the last column, which is very tiny font and very dim, you have to trust me, there's tons of zeros, which is how many out of 10 papers in each of these journals actually adhere to these policies. Recently, we published another paper in PLOS Biology where we looked at a random sample of PubMed in the last 15 years. Out of 500 papers, 259 had empirical data. How many of those shared all their data? Any guess? Maybe you have read the paper. Of 259? Quote? 1. You're an optimist. Actually, 0. But there was one that shared the protocol, and actually that paper was a protocol. The protocol was a separate paper. The incentive was that you can get a publication if you share the protocol. So there's plenty of room to improve, but then who's going to do that? We have all these PIs struggling to meet deadlines and submit more grants with 6% success rates, and now you're asking them to share all the data. I got an email Thursday evening about sharing all the data from a paper that I recently published. And that person, who's a wonderful scientist, wanted that information by yesterday, practically. So by Friday morning, the data have not arrived yet. And we were working over the weekend to try to share that information with him. So there is a lot of difficulty about who is going to do it, with what resources, when, who can really have access to these data? Everybody. Who will be able to transform these data sets into something that would be usable and not lead to further misleading analysis if people don't understand what the variables are and what the code books mean, or if there's no code book, even worse. And then you can really create even more noise than try to save some of these problems. Here's one example of raw data that have been used and have been published. This is an evaluation of all the clinical trials that we could find in PubMed where someone had reanalyzed the raw data for the very same hypothesis that was proposed up front. We found 37 such trials, and for the very same clinical question, in the same population, same exact data set, 35% of the time the conclusion was different on whether patients should be treated or not treated, or who are the patients to be treated. Who did these analyses? Maybe one might think they were rogue analysts trying to put the original investigators into shame, their enemies, and actually it was the same investigators who published the first paper. And why would they do that? Well, because in the current environment, if you waste 3 times to perform a reanalysis and you get the same result, who's going to publish that? It's going to be a very tough sell. A few of them did get published, but I'm sure that many, many more were done. Conversely, if you can recast the conclusion, oh, I told you that these are the patients to be treated. No, forget it. They should not be treated. Now you have a new message. It's a confusing message. It's the same people coming up with that confusing message. But the current incentive system is actually encouraging this. The incentive system is also involved when it comes to how research should be sponsored. So some people clearly have strong conflicts with the research and the results to be obtained, or the interpretation, the reproducibility of inferences. So one might wonder, who should sponsor randomized trials? Who should sponsor meta-analysis of randomized trials, or cost-effectiveness analysis, or guidelines, or other sensitive studies? Should manufacturers be involved, or not involved at all? To me, I think we should try to detach them as much as possible from that process. Here's one example. We looked at 57 trials published in 2011 that had a head-to-head comparison of a sponsored drug, where the manufacturer was sponsoring the trial, versus another drug. And 55 out of 57, the conclusion was favorable to the manufacturer. That's close to 97% success rate. I would say that we could have skipped these trials. We would have said the result is nice, it's favorable, don't run them. It's probably a preset conclusion that these trials were aiming to get. Meta-analyses are not exempt from that problem. This is another analysis that we published recently, where we looked at all the meta-analysis on depression that had been published in a period of 6.5 years. We found 185 such meta-analyses. That's amazing. That's a single type of drugs for a single type of indication. It's a factory. About 80% of these meta-analyses had some involvement by the industry, and a good size of those were actually co-authored or authored entirely by industry employees. When we looked at the conclusions, whenever there was an industry employee co-author, then always, with one exception, 53 out of 54 times, there was no caveat about antidepressants. Conversely, when no industry was involved, the rate of caveats was about 50%, about no effectiveness or severe toxicity or both. Actually, I'm not sure whether that one that found caveats and had an industry employee, whether these people are still employed by the company. So supposedly meta-analyses are the top of the hierarchy of evidence, but still they can be manipulated in terms of their conclusions. Better statistics and methods. I can spend hours and hours on that. We have lots of empirical evidence that suboptimal methods are used. This is just one example from animal experiments. How often do they use randomization? About 20% of the time. It's something that could have been used standardly. How often do they blind investigators reading the results? About 10 to 20% of the time. Again, that's something that can be done at no cost and with much better reliability of the results. Standardizing definitions and analysis. Lots of observational research has no standardization at all. This is looking at a snapshot of some of the studies on pterygium risk factors, and I'm showing you the risk factors that everybody practically agrees in high expertise reviews that they are important risk factors of pterygium. I'm just showing you how many of these studies adjust for these risk factors. None of these studies adjust for all of those. There's not two studies that adjust for the same set of risk factors. Everybody's doing it in a different way. Even though there's consensus, there's complete lack of consensus on how the analysis is going to be run. What happens if you can run analysis in many different ways? Then you get clouds of results instead of a single result. And this is what we did here. We took data from NHANES, the National Household Survey, and we took 4 factors, vitamin D, thyroxine, creatinine, and alpha-tocopherol, and we tried to ask the question, is the levels of that factor, are the levels of this factor related to death risk? So you get these clouds of results that have the hazard ratio on the horizontal axis and the minus log p-value on the vertical axis, and there's about 1 million points in each one of them because we have run 1 million different analyses. How do we do that? Well, along with these factors, there's other things that can affect the death risk. Men or women, age, other diseases, exercise, nutrition, whatever. You can start adding those or not adding those. For each one of them, you have 2 choices, to add it in the model or not add it. 2 times 2 times 2, if you go to 2 to the 19th, you go close to about 1 million. And in many cases, you get this Janus phenomenon, this V shape. So alpha-tocopherol, depending on what you adjust for, you can get the result to be very nice, so it decreases mortality, or you can get it to be very bad, it increases mortality. And this is just playing with adjusting risk factors. If you do that for other modes of the analysis, you can probably get a Janus phenomenon looking in both ways for almost any risk factor in observational epidemiology. So we need to live with small and tiny effects, and we need to forget about big discoveries for a while. It would be great whenever we come up with big discoveries, but across 250,000 trials, when we looked in 85,000 meta-analysis, we could only find one intervention that decreased mortality 5-fold and could be seen in 2 trials and had significant results. So most effects are very small. When you see a big effect, when you try to replicate it again, it becomes small. We can improve reporting. The Equator standards, you can go to the Equator site. There's 200 standards roughly of improving reporting of research. We can do more research on research to see whether some of these practices work or not. There's a lot of that happening. Just in 5 months in 2015, there were more than 800 papers looking at research practices and evaluating empirically or with theoretical constructs the way that we perform research. Our new center at Stanford is trying to be a connector hub for such efforts. So if you're interested, please do get in touch. I'd love to hear more ideas and more thoughts. And I will close with 2 slides about how some of these solutions may be aligned with our incentives and rewards. Currently, we're mostly rewarding people for publishing significant results, as I showed you up front. And this is just the P of the PQRST electrocardiogram of reward of incentives. So we reward productivity, but we need to reward also quality, we need to reward reproducibility, we need to reward sharing of data and other resources. And for those people who do applied research, we need to find ways to reward the translational impact of the research. How would we do that? Well, we can change the value that we attribute to different aspects of what we do and what we produce and what we communicate to other scientists and to the community at large. So currently, there's practically no value in sharing data, protocols, analysis codes, contributing in important ways to peer review, contributing to educating other scientists to get them to become more reproducible in what they do. What matters is publication. So there's 1 point for a good publication and grants. This is 5 points for an R01. And then with those, with that money equivalent, you can buy other wealth items. So you can become an assistant professor, associate, or tenured. If you have postdocs, this is 2 points per postdoc, which means you can trade 1 postdoc for 2 papers, or vice versa. And if you have administrative power and you're a good lobbyist, you can go up to getting 200 points for free. So how do you change that? One option is that you give no value to publication, but you give value to replicated publications, and you give even more value to successfully translated results, and you lose points if something gets refuted. You give value to sharing, to peer review contributions, to education contributions, and so forth. Or you can be even more disruptive, which means that maybe people would lose points when they get grants. Then people would not want to get grants because you got a grant, you better do something with that. You got all that money. Or if you have power, again, you lose points because you have all that power. You'd better put it into some good use. I'm not saying that the last column is the best that we can do, but we need to think about what we are rewarding people for and to find the best solutions on arriving at the most desirable results. And we should also not forget that it's not just scientists in the game. Many other stakeholders who are interested in science for different reasons. There's the industry, including both sales and marketing and R&D. There's universities, institutions that perform research, entrepreneurs, philanthropists, hospitals, insurance companies, state and federal authorities, non-scientific staff, consumers, people who, especially for health research, they want to remain healthy or to avoid disease or to cure disease. And their priorities are different. They may want to publish. Lots of scientists, we want to publish. Fundable research may be the ideal for others. We want to get funding for whatever we do. Translatable research is important to patients and many other stakeholders. And some stakeholders want to make money. They want to profit out of that. So we need to find ways to align the interests of these stakeholders. I think that I have shown you some empirical evidence about reproducibility. As I said, this is evidence that is accumulating. Some of that may not be reproducible, and I think that we need to do more of that to be sure how much of that is reproducibility affected. The available evidence suggests that there's substantial room for making improvements on the way that we perform research, on our research practices. However, as I said, some of these suggestions have not been very well validated, and I think that we have to be careful in trying to translate different solutions to different fields. I think that eventually, as we have this wonderful success of science with millions and millions of papers and millions of databases becoming available, we have a wonderful opportunity that we can look at how we're doing and go in a self-enhancing mode and create more reliable results. Eventually, I think this will be the legacy of the science that we are contributing to other scientists and to our communities at large. I'd like to close with listing a few of my collaborators who I have worked with in some of the empirical data that I showed you today. Without their contribution, clearly, none of what you saw today would have happened. Thank you.
Questions, I'll ask you to put up your hand, but then wait patiently for the microphone to come to you. So,
first, let me say, one of the first things that you learn as an economist is there's no free lunch. And I always think that your work is maybe an exception for this. It may be a magical way of improving the value of science at very low cost to the system. How much value by doing statistics wrong? I want to spread my admiration for what you're doing. I have a question, too. You're thinking very hard about solutions from an institutional point of view. But still, after many years of having opened the well of this, the way you present things is around hypothesis testing. And for example, in your last slide, you talk about rewards and punishments for getting it wrong. The question is, isn't there a more fundamental problem, which is about what is research and what is knowledge, how we should think about this, which is the basic idea is you do an experiment. You're trying to figure out something about nature. You always get noisy data. And at the end of the day, what every experiment, what every studies give you some observation, likelihood information, some evidence that may lead you in the right or the wrong conclusion, but it's only when you pull all of those things that you can really understand. So in some sense, as long as the statistics and the likelihoods are reported correctly, there is no correct or incorrect. You're adding to the pool of knowledge, and you should just reporting, this is where my evidence is pointing me to. That's it, not this is true or this is false. Why not take that next step?
I fully agree. I think that the dichotomy of true or false is problematic in many areas, because you're more interested about the cumulative evidence. And I would argue, nevertheless, that this poses an even stronger incentive for making the complete evidence available. Because if you're interested about the cumulative evidence and you have some selection force that allows only some of that evidence to be disseminated or become visible, or only particular types of results are considered to be interesting enough to be communicated, or people have to modulate their analysis or their protocols in order to obtain these desirable results. And then obviously the cumulative evidence is not going to solve the problem of lack of reproducibility. It will only create some seemingly reproducible chain of evidence that actually is just entirely wrong. And we have seen that repeatedly. I mean, we've seen many situations where some false notion was entrenched in the literature. Somehow, famous experts supported it, and then people continue to build on that, having the expectation that their results need to fit to that paradigm. And it took a very long time for that whole cumulative chain of very selected results to be dismantled.
You mentioned changing the economic incentives of, and basically changing the weights of the incentives. So suppose that we do change these weights, then what's to stop another organization from having a different set of weights, and how do we determine which weights to use? Because in today's world, there's so much competition. If you start like a new system, then you're going to have like 20 other people start a new system. Right. So how do we determine which one?
So I think that based on the solutions that we have seen so far that have worked, it's not easy to change a system by imposing some new weights, kind of, in a dictatorial fashion that, this is how you're going to do things from now on, you will get lots of pushback. And I think that the pushback will actually be reasonable because this is not how science has grown over time. Science has grown more in a grassroots way. So, scientists accept that I need to do that because if I don't do that, then I will waste my time, I will waste my life, I will waste my money, I will waste my resources, my postdocs, and I will get nowhere. So, for example, what happened in psychology, we saw a field that changed from no replication ever being desirable to most people really thinking of replication very highly and believing that this is the way that they need to do their work. Or what we saw in genetics was a field, again, that no replication was done and no sharing was ever happening, to a field where everything was happening now in a collaborative mode with large consortia and with sharing. So it was the scientists themselves who understood that that's the way to make progress. And I think the same applies to almost any of these changes. I'm not proposing that someone should impose a change without the scientists understanding that this is really to their benefit. Now, if you want to make changes, you need to have buy-in. I mean, you cannot just say that, okay, here's one team who's going to do that. Maybe they will seem interesting to outsiders. They will be kind of a peculiarity, but this may just die off. Conversely, if you have multiple stakeholders supporting that, it's likely to be successful. So registration is something that had been discussed for 40 years. And it was not happening until not only scientists, but also all the major clinical journals said, we're not going to publish a trial unless it's re-registered. So then if you wanted to publish a trial in a major journal, which is the very old system of productivity, publication and impact, you had to fit that rule. And people agreed that this is a good thing to do. It's a different issue that's not perfectly applied, but at least it did happen. I mean, well, it was just a dream in the past.
Again, when you mentioned the changing the weights of a system to produce the more reproducible results and sort of change the institution to make the investigations more effective, sort of when you mentioned the grassroots evolution of science, I feel that the way science has evolved, for instance, to place emphasis on funding, perhaps while not ideal, it's almost a functioning system. What sort of alternatives could you see that could also function in a similar way? For instance, if you were to place less weight on gaining funding, where would science then find a replacement for such a system? Where would there be money for more science, for instance?
Yeah. I'm not saying that one should decrease funding for science by any means. Quite the opposite. I mean, science is the best thing that has happened to humans, and any funding that you can get to support it and increase the funding is a wonderful idea. The question is, how much do you prioritize, for example, research careers, or you funnel resources based on, let's say, a prior funding success history? It may be that in some cases this is not a bad criterion, maybe. But we have empirical evidence that in many cases some of the brightest scientists who are more out of the box and innovative thinkers, they don't fit necessarily to some types of funding systems. There's many different types of funding systems. They have very different criteria. We know that there's empirical studies, even experimental studies, that show that there is a funding bias against out-of-the-box ideas. We know that even in the best functioning systems, like Australia and Canada, and some evidence also from the US, about 2/3 of the variance in funding decisions are random noise. It's not completely random noise, but a good size is random noise. So I think one doesn't need to be disruptive and say, oh, no, we're not going to have funding for science. Right? I think that would be completely wrong. These are research questions. These are questions that can be addressed with research studies, with some observational studies, but also with some experimental studies. So currently I'm collaborating with scientists in many funding agencies to try to come up with some such studies that can test some of the processes and see whether they can be improved.
In the case where there might be an industry partner in the research, such as a pharmaceutical company, and how a lot of times the research might yield the right answer. So what is a suggestion there? And do you see that with the types of registrations that you mentioned be sufficient for such a case? And do you see it getting to a healthy point where there's an avenue for, say, pharmaceutical companies to do a validating research that's trustworthy in the community?
So my personal opinion about that is that there's some misalignment of who is doing what. So currently, the pharmaceutical industry is using research from academic institutions, the preclinical part, the translational part, and they try to develop the licensing evidence for these products that they develop. I think it should be probably the opposite. So I think that the translational part that delivers a new drug or a new biologic or a new test to be used diagnostically or in screening or monitoring or whatever, that part should be funded by entrepreneurs, the private sector, the industry. Because they have every reason to do the right type of research and ask the right questions and invest their resources in the best possible way to create something, to deliver a product that will work eventually. The testing, the licensing, and the proof that, yes, this thing does work and it saves lives, it decreases clinical manifestations, it's better than something else that we have. I think that part should be funded by public sources. So the way that I see funding, at least in biomedicine, is blue sky science, public, because who's going to fund it otherwise? This is just knowledge, no deliverable practically. So public funding for blue sky science, entrepreneurs, private sector, industry for translational, deliverable-oriented research, and public funding again for randomized trials proving that this thing does work in real life. It's almost the opposite. I mean, we do have public funding for blue sky science, but actually shrinking, unfortunately. Most of the translational is done with public funding, and almost all of the evaluation and licensing is done by companies who have to show that their drug works. So I can't blame them. You're asking yourself to show that you're the best painter, so yes, my painting is the best.
So I would agree that science has to be reproducible ultimately in order to move forward, but there seems to be, maybe I'm misinterpreting, an implicit assumption here that if there's a study that is published that produces a result and another study that doesn't replicate that result, that it is the initial study which is wrong, flawed, and I don't see that that's necessarily the case because it assumes equal competence on the part of the replicator and the originator, and particularly in experimental biology, there is implicit knowledge, there are uncontrolled variables that can often make the difference between successfully executing a certain experimental protocol and not being able to replicate a protocol. A classic example of this is in the early days of recombinant DNA. Within a single laboratory, there was one person who was able to prepare reproducibly libraries of cloned DNA and other people who could not do that. The successful person was very sloppy. It turned out the reason they were successful is because the enzymes used to create the libraries were very labile. And so the faster you worked, the better the chance the enzymes would be active long enough for the reaction to conclude. Those who worked carefully, methodologically, and slowly failed to produce the libraries because by the time they cooked everything together, the enzymes were dead. So I would just like you to comment on this issue of, just because something is published and not reproducible, that is really the original study that is flawed, and how do you factor in your analysis these issues of implicit knowledge and uncontrolled variables, particularly in sciences that involve a great deal of, I would say, cooking work, as it were.
I fully agree with that. So, and this is why I had that 3x2 table, where it could be that the original study was correct or wrong, and the replication could be correct or wrong. So, you can have all combinations. You can have the original study being correct and the replication study being wrong for whatever reasons. And this is a very nice example that you put forth. And I think that this may be more prevalent in some fields compared to others. So there are some fields, and this is why I started with that granularity of different disciplines having very different rules and very different requirements. There are some fields that practically it all boils down to a 2x2 table. And it's a very crisp, concrete measurement. There's no intervention, no experimental magic or uncertainty or uncontrollable or unknown parameters. You just populate that 2x2 table and you get some result and you can compare, and then it's a matter of who has the most weight, other things being equal. In some cases, it could be that you have far more complexity, lots of factors that are not easy to control, may not even be visible at the time that the experiments are done. People may not be familiar with what are the important aspects of how to do the experiment. In these cases, I would say that replication is still important because it can give us a sense, well, it didn't work. Why didn't it work? And I have to go back and start cataloging. There's 5 potential reasons, and then start working through the reasons. And then we identify that one reason that was really the most important that perhaps was actually missed in the original experiment. So the original experiment was correct, but the people didn't know why they got that correct result. And once we find out that this is the reason, then we can not only repeat that experiment again, but we can use that technology in a more generalizable way, and we can build other experiments based on that. So there are fields that have this sort of complexity. So one has to be very cautious about saying the first study is judged by the second. I mean, all studies are judged by all others, and they offer some complementarity at the end of the day.
Hi, I have 2 questions that are about the policy implications of what you're talking about. The first is, so reproducibility sounds great, but do we actually have any evidence that it actually matters for real-world outcomes? I mean, so at one point you mentioned there are some areas of science where, if you give the wrong drug, people die, versus others where, you mismeasure how long a toad's, toe is. Someone will figure it out later on. It doesn't matter that much. So that's one thing, but that's actually different than, do you have a sense that more reproducible fields are either more profitable, deliver better drugs, cure more people, anything like that, right? So there's that.
Yeah.
And the second question is related. So you attributed many to the problems to the fragmented and small shop nature of science, right? So if so much of the problem is about inadequate sample sizes or underpowered, many of the problems you are linked to. And then the solutions are about sharing of data and pooling of results and collaborating on what our problems are, and then sort of pooling ideas and not having individual small shop grants. Well, is it worth the trade-off to have less science on fewer topics that are more robust? And that seems to contradict what you said before about, we should fund blue sky science, right? Blue sky science is the stuff that's going to be underpowered. So how do we reconcile these questions of less science that's more robust? And
Both questions are really great. So the first one, you need to look at fields that are in the late translational phase. So, on applications that are about life and death and, like medical intervention. So Vinay Prasad has done some very nice work looking at medical reversals. So about 50% of medical practices that have been long established, when we tried to examine them in rigorous ways with well-done randomized trials, they don't work. So, these are things that we have used all along in millions of people. And there's many other empirical evaluations that show kind of a gradient of reproducibility as you move from interventions, medical interventions that we have documented with large trials and with pragmatic patient-relevant outcomes, to smaller trials that have used surrogate outcomes, to observational research that has supported some interventions or some of the recommendations that we make. And the gradient of success and reproducibility goes from 90% down to 30%, down to 5%. Stan Young had published a paper looking at 52 claims about lifestyle and what to do with your life based on observational evidence, and none of them were reproduced. So we do see that there is quite a big gap of reproducibility, even at that late chain of do things work. Now, about giving lots of options and lots of choices and lots of opportunities to do different projects with different ideas and let many flowers blossom and then we get some of them to be the best. I like that. This is not really what's happening when I'm talking about little shop with small sample size. Most of these small sample size studies are not really different. They're not different flowers. They're the same flower pretty much, maybe a little bit different. I mean, everybody's trying to say that what I do is it's different compared, but actually it's pretty much the same. And we see that in very different types of research, from early discovery all the way to applied clinical studies. So to tell you a little bit about one end of the spectrum, there are some drugs where we have more than 1,000 trials out there. And we don't have a single trial that would be informative on its own on whether it works or not. I think this is unacceptable. It's not that I'm limiting the creativity of anyone. I mean, these are drugs that we just need to know, do they work or not? And what we have is 1,000 trials spread thin all over the place with 10 people there and 10 people here, and we'll never get an answer. Nobody has used a good clinical outcome for these trials. The same thing applies to earlier stages of research. So all of the genetic epidemiology that I showed you, people were doing the same thing for 20 years. They were all studying the same genes and the same phenotypes and getting nowhere until they decided to join forces, and they said, we're doing the same thing. Let's acknowledge that. So by doing that, I think you can probably liberate, free more funds to really allow more flowers to blossom for people who really do different things, completely different things.
Is a little bit different. I'm Elaine Bearer, and I'm an editor for the PLOS journals, and I think that the journals play a big role in this. So we've been meeting and discussing how could PLOS journals improve the situation, and one of the things that's coming out is that we want to make sure that all of the papers we accept publish their complete protocols. So when you publish in Science or Nature, you're constrained. You can only publish a tiny amount of what you actually did, and that's a huge problem for reproducibility. So I think that all of us who are reviewers and editors could play a role in this by insisting on when you accept a paper that the complete protocols are published. The second thing I wanted to say is that there's one thing I think is missing in your list of reproducibility, and it's something that as a molecular biologist, and I know David worries about this and does it. We always try to do another experiment that would answer the same hypothesis, but doing it in a completely different way with a different method. And by doing two different methods, asking and posing the same hypothesis, we have a much better chance of having a reproducible, biologically significant result. And finally, I want to say, I think statistics is taking way too big a role in all of this. And I think if you don't have a good effect size, statistics doesn't solve it for you.
Yeah, I think that all of these comments are very accurate. I think that journals do have a very important role because they are a key rewards distributor in a way. And I'm very much in favor of making protocols available. There are some caveats with that. So, for example, what is a protocol and what version of the protocol are we talking about? So we see that very often in clinical trials, which supposedly should have the most robust protocol possible. I mean, it's not that it's something that is exploratory and it's evolving over time. But even in clinical trials, you may have 10 versions, 10 amendments. And most of these amendments may happen at a time that people amend the protocol without knowing the results so that they might influence the results of their interpretation. Other times, maybe this has happened after some insight about what the results are. So there's many amendments and evolutions of a protocol that have very different connotations about whether the result eventually is both transparent and reproducible and not affected by experimenter bias. When you go to other types of research that are more exploratory and more iterative, and people may have tried lots of bifurcating paths, as Andrew Gelman has proposed, then I would argue that we need to see as clear a picture of what these bifurcations have been. But it's not easy. And I say that for my own research. Whenever I'm in exploratory mode, can I go back and explain exactly what I did in each one of the decisions that I made on how I wanted to approach that question in the next round. So I think much of that will become visible as we get more experience about what protocols mean and how they look like and how they're judged by colleagues and the community at large. And the second question was about the triangulation type of approach, which I'm very much in favor of. The only caveat with these corroborative replications or triangulations is that you need to find ways to verify that there's no experimenter bias involved. So this is a situation where blinding of the investigators to the results, for example, in reading the results, and some other safeguards are very important, because otherwise, experimenter bias, since described by Rosenthal almost 50 years ago, it's very strong. I mean, we want to see a pattern. I mean, once we have seen that, we want to see it in some different experimental system. And that can easily get things astray. So I'm very much in favor of that, but it has to have the proper experimental safeguards. And these depend on what type of experiment that is.
Hi. That was a great talk, and I can say that I have some contradictory feelings. That's actually, it's very rare when I go out from a talk, okay? And I want really to share with you. I don't have a specific question, but I want to share these contradictory feelings that I have. So I'm doing research in neuroscience, and I'm doing both computational and mathematical modeling to understand human and animal behavior, but I'm also working with non-human primates, with monkeys. So I will feel comfortable to share my math and my computational models, but I'm not sure, although I can see your point, to share my monkey's data while I'm doing the experiment. Because for a monkey's data, for a monkey study, it takes a very long time and a lot of pain and effort to get just a single publication out of that. I can tell you from my experience that it took me about 4 years to train 2 animals in order to start recording from them, just like starting to do my experiment. And I'm pretty sure that my PI also has the same feeling with me. And I can tell you for sure that all people that were doing this kind of research, they're a little bit skeptical on how, even though I can see your point that we should share the data, and I agree with you, and I can go one step forward and say that imagine for me spending 5 to 6 years to just get out one single publication and then having the data sitting on my desk. And nobody else is going to use it. I really want to share the data, but on the other hand, I don't want to have people to take advantage of that by saying, I'm not going to do anything, I'm going to wait for him to finish his experiment, and then I will take his data and publish another paper. Just quick and dirty.
You're right. I mean, this is a very realistic approach to some dilemmas that arise on a daily basis. And I think that some fields have more of that problem compared to others. I think it depends on whether in your field there's some security that if you put these data out in public use, and let's say someone else is going to use them somehow, will you get some credit for that? So if you get credit, and let's say 10 other teams use these data, but you get credit, and to your community, it's as if you have really been the cause for these other 10 papers. I would argue that it's a clear advantage, because you worked for 4 years to get that one paper, and now, because you shared that information, you get the equivalent of another 10 papers in terms of how the field is seeing you. But if the field is saying, he was so stupid, he did all this work and he just gave it away, obviously, it's not going to work. So the field needs to reach some understanding on what it means. And I think that the people who work in each field are in a better position to understand how much effort it means to do this part or to do that part, and how important your contribution would have been. Given that there are fields that have shared information widely and that also put a lot of work, I see no reason why this cannot happen, provided that there is that transparency and that sense of justice. Because as you say, otherwise, you feel like I'm a fool to do it.
Make a very similar point. You mentioned earlier that people work in silos many times, and reproducibility at different levels. So if we could start getting credit to produce good data, same as being able to publish results from it, that will be a great thing. So I work in astronomy, and there, for instance, some of our groups, including ours, we put out our transients in real time without any classification involved. So that is like publishing only without any conclusions, without publication. Credit is given for that, and that's looked at good. But there are other groups where they say, no, if you're not doing classification, then that is not science. You should not really be doing that. And even in the biology, actually, if one can start attaching significance to producing, then that is one of the levels of reproducibility that you mentioned. And so there are now methods available where you can attach programs to your papers. You can attach code that makes figures out of that. And if that goes on, that will be fantastic. So,
yeah. I think that there's many examples that this can be made to work. And I mean, you mentioned one example where sharing the data set is one act of academic credit, in a sense, on its own. And then you can build on that. You can build adding information, software, code, observations. It's probably less common in the biomedical sciences as opposed to physical sciences, but I think we will see more of that. So there's one effort like Science Matters that is practically aiming to publish single observations. So instead of waiting to build a full story of 20 experiments and observations pieced together in a full narrative about here's the whole pathway and why this is important, and I have triangulated in very different ways, you just publish a single observation that's peer-reviewed, of course. When and how is to be discussed. And then you can build, here's the second, third, some other may add a fifth observation, and you see that community of observations growing and building that more complete narrative eventually, but not necessarily done as one step. So I think that we need to experiment with different approaches, and some of them may work very nicely in particular types of investigation.
I would just like to say that you focus on a lot of big data statistics. Is this reproducible? Is this not reproducible? But that the idea of the literature is to address other experts. Possibly those experts know whether or not this is likely to be true or not. So even if you say, okay, a lot of these studies aren't really reproducible, is it possible that an expert in that field wouldn't be so affected because they know, like, this person, this type of study that they did, they'd give it less weight. So it's not, you looking at it objectively, you see, oh, there are all these papers that don't reproduce, but someone in the field will say, I can sort of tell what papers are good and what papers aren't.
Yeah. It depends on the field. I think what you described was a fairly common scenario, let's say 20 years ago, in most scientific fields. Because most scientific fields had a limited number of people working on them. And there was this grapevine knowledge. People would share information at meetings that, oh, I tried that and it didn't work. Even though it's a cell paper, it's not working. Or yes, I could do that, or I built something on this and I took it to the next step. So in fields that have small membership and you have these experts who kind of have that view of the field, and whatever is not published they also know through their discussion and connections and communication with other experts, this can happen. But there's many fields, and most of the fields that publish the largest number of papers nowadays, there's no single expert that can have even a small portion of that knowledge. So it's impossible to know what is being done in 300 teams or 500 teams or 1,000 teams around the world who may be working in that field. So you just then base your inferences based on gut feeling. And this is not very good judgment. Sometimes the gut feeling is very easy to have very strong gut feeling. So I remember in my early days, I was working in HIV research, and I was at NIH at the time where we found that triple antiretroviral therapy could really change the course of disease, and people who were dying of a lethal disease now would have a chronic condition and live almost normal lives. So it was a completely paradigm change. But prior to that, for a very long time, we were working with treatments that really didn't work. And at some point, I did a systematic review of all the treatments that I could find for HIV infection before we found some really good treatments. And based on the literature, all of them worked. And all of them decreased mortality enormously based on the published information. But we knew that people were dying. I mean, they were dying in my clinic, they were dying everywhere, and we could not really affect that based on the ineffective interventions that we had. So here you could easily say, oh, no, that's not so. You can just forget about it. But in most fields, it's not so transparent. You don't have 5 million people dying all around you in the world. Usually it's some knowledge that has no immediate translation that you can base your intuition upon it.
All right, thank you all for your questions, and let's thank Dr. Ioannidis one more time.